Good afternoon. I'm Amy Lensing-Tate, the Executive Director of the University of Wisconsin-Milwaukee Alumni Association. Welcome to all the alumni and friends across the country. Our mobile Masterchat series provides a snapshot into the world-class research taking place here on campus. Today is certainly no exception. We'll hear from UWM Distinguished Professor of Psychology, Dr. Karyn Frick. Dr. Frick is a behavior neuroscientist and a neuroendocrinologist. Also an adjunct professor of pharmacology and experimental therapeutics at the Medical College of Wisconsin and the chief scientific officer of Estrogen Therapeutics. She has nearly 30 years of experience studying the neurobiological mechanisms underlying memory formation, with a particular focus on the cellular and molecular mechanisms through which estrogens regulate memory consolidation. She's the editor of a book, Estrogens in Memory, published by Oxford University Press in 2020. She's a fellow of the American Psychological Association and Association for Psychological Science, and is also the president-elect of the Pavlovian Society. Prior to being recruited to UW-Milwaukee in 2010, Dr. Frick was an assistant and associate professor in the Department of Psychology at Yale University. I will join you again at the end of the chat for questions, so please put those on into the chat window. But now it's my pleasure to turn things over to you, Dr. Karyn Frick.
Thank you so much, Amy, and I very much appreciate the chance to talk to alumni and give you a sense of what's going on in my lab and in the Department of Psychology currently. I'm going to share my slides now. Let me just bear with me while that happens. And hopefully you can see those. There we go. Okay, so as Amy mentioned, I did have a book published in early 2020 on estrogens in memory. And I've been studying estrogen in memory for about 25 years now. Indeed, this has become a growing part of the field of neurobiology of learning and memory. And this book contains 27 chapters by leading scholars throughout the world that cover this topic, from everything from cellular molecular mechanisms up through human clinical trials. So it's become a very interesting topic to study that has garnered a lot of interest lately. And before I get into the meat of what I'm going to talk about today, just a little bit of background about how I came to UWM. Sorry, my screen is not advancing. There we go. Because I'm not native here to the Milwaukee area or Wisconsin at all, or even the Midwest. I'm from New Jersey originally. Earned my bachelor's degree at Franklin Marshall College, a small liberal arts college in Lancaster, Pennsylvania in 1991. Which is actually where I met my husband at Franklin and Marshall. My husband, David Frick, is a professor in the chemistry and biochemistry department here at UWM. So we have traveled through academia, been fortunate to travel through academia together all of these years. After graduating from F&M, we then did our PhDs. I also got my master's at Johns Hopkins University, for me in the Department of Psychology, and then did a postdoctoral fellowship in biological sciences at Wellesley College, where I spent 4 years before moving to my first faculty position at Yale University, where I was an assistant and associate professor. Was there for 10 years. And the work that I will talk about today really started when I was a postdoc at Wellesley, really expanded when I was an assistant professor, associate professor at Yale. And then in 2010 was recruited to UWM to help sort of boost our burgeoning neuroscience program in the psychology department. So I've been here moving through the ranks for the last 10 years. And I just want to say a word about the neuroscience area in psychology. There are currently 11 of us who study largely the neurobiology of learning and memory. It's one of the rare groups throughout the country, or even in the world, that's really focused on learning and memory as its major research interest. There are some of us who study neurobiology of emotion, as well as childhood neuropsychiatric disorders and stress. I want to highlight two of our newest faculty hires. We were very fortunate to hire Arun Asak and Caitlin Bowman just before the world closed for COVID. We hired Arun and Caitlin in February of 2020, and they both started in January of 2021. So they are really brand new faculty, but we are absolutely delighted to have them. Arun studies the neurobiology of memory in mice, very similar to, with similar questions to my own lab. And Caitlin Bowman studies the neurobiology of aging and how aging changes, cognition changes with age in humans. If you're interested in learning more about neuroscience and psychology, I'd encourage you to visit our website. So, as I said, I've had a long-standing interest in the neurobiology of memory, as do my colleagues in psychology. And I'm interested not only in what happens when memory formation goes well, but what happens when memory formation doesn't quite go as planned, as often happens in aging and neurodegenerative diseases like Alzheimer's disease, as well as the topic we'll spend much time on today, menopause. And my work in the last 25 years or so has been motivated by this particular sex difference. Well, really sex differences in the rate of memory decline in aging, whether that's normal aging or dementing diseases such as Alzheimer's disease. So that sex difference is reflected in this figure from the Alzheimer's Association, showing the estimated lifetime risks of developing Alzheimer's at ages 65, 75, and 85, with men in the purple and women in the gold. And you can see that at every age, the risk is approximately twice as much in women as it is in men. And this is taking into account the fact that women live longer than men. So aside from age, which is the greatest risk factor for Alzheimer's disease, female sex also is a major risk factor for Alzheimer's. And we are trying to figure out why that is. One major contributor, we think, is menopause. And as I'm sure you're aware, hormones change significantly during menopause, particularly in the menopausal transition. So this graph shows you increasing age from age 20 to age 70 with sort of optimal levels of estrogen and progesterone levels that are in women, optimal levels are high levels. This is post-puberty. The female body and brain are bathed in fairly high levels of estrogens and progesterone as a result of the menstrual cycle. Those hormones are produced by the ovaries in response to signals from the brain. And so levels remain, even though they cycle, they still remain fairly high throughout the early adulthood, and then towards middle adulthood in the 40s. You can see this precipitous drop in estrogen and progesterone levels, such that levels are quite low in the 50s and beyond. And we think that this is important for cognition because it turns out that while these hormones are obviously very important for reproduction, neurons in the brain, so neurons are the main communicating cells in the brain, neurons come to depend on these hormones, particularly estrogens, for proper functioning. So estrogens are what we call trophic factors. These are chemicals that help to help cells function well, function properly. And so during middle age, we see changes in the brain as a result of, sort of long-term use, cellular damage that accumulate as we get older. So we have this backdrop of changes that are happening in the aging brain. And then for women during the perimenopausal period when hormone levels plummet, we take this important trophic factor that helps keep those cells functioning properly, we take it away. And we think that the loss of this important nourishing trophic factor renders those neurons more vulnerable to the detrimental effects of aging and neurodegenerative diseases like Alzheimer's disease that cause numerous cellular changes in the brain. So this would seem like a fairly simple problem to solve, because if low estrogen levels result in increased risk of dementia, that we could simply replace estrogen levels and reduce risk of dementia. But it's not so simple. Hormones are complex chemicals that have receptors throughout the body, not just in the reproductive organs. And so you surely heard reports in the news about the side effects of hormone therapies. So I'll start with the good. There are many very beneficial effects of estrogen therapy, including strengthening bones, improving cholesterol profile, and particularly for reducing menopausal symptoms like hot flashes in women. They're very effective for that. But there are small but statistically significant risks of negative side effects like breast and endometrial cancer and heart disease and stroke. And so millions of women have stopped taking hormones because of those increased risks of cancer. And they're left frequently with unproven biologicals or dietary estrogens. But we know that estrogens have really beneficial effects, as you can see on things like bone health and so on, but also on the brain. And so we think in my lab, we are trying to figure out exactly how estrogens, like the one I'm showing you here, which is 17-beta estradiol, and you're going to see that as abbreviated as E2 in several slides. How do estrogens regulate memory? Because we think if we can understand on a cellular and molecular level how this hormone regulates memory, then we can develop drugs that do the good things that estradiol can do for memory without some of those detrimental side effects. So if we can, look at the effects of estradiol in a brain region that's important for memory, like the hippocampus, which you can see here in the warm colors, the orange and yellow colors, how that leads to a memory, we can better design drugs to provide benefits for memory. So I'm going to cover, our approach is sort of three-pronged. We first determine how estradiol regulates memory in young animals. How does estradiol regulate memory in an optimally functioning system, which is young adult animals? We then can take those lessons learned and try to understand why hormone loss leads to memory dysfunction in models of menopause, in aging, and Alzheimer's disease. I'm not really going to get a chance to talk about that today, but I'm happy to answer questions about it because I want to get to some of our work taking the lessons we've learned and developing new treatments for menopausal symptoms. Before I get into that, though, I want to address some myths about sex steroid hormones that are common in the media. So first of all, sex steroid hormones as a class are all derived from cholesterol. So cholesterol gets obviously a bad rap. High cholesterol is bad, but we still need cholesterol because cholesterol is the precursor for all of the sex steroid hormones. And by sex steroid hormones, I mean progesterone and its metabolites, including testosterone and other androgens, and all of the estrogens. So progesterone, testosterone, and the multiple estrogens. So here's one thing I want to make clear is that there is not just one estrogen. We frequently hear people talk not only in the media, but in science about estrogen. Estrogen does this, estrogen does that. There is no such thing as estrogen per se. There are numerous estrogens, of which you can see 3 of them here. Another thing is that you can see that in order to make estrogens, we have to make androgens like testosterone. So there's no such thing as testosterone being a male hormone or estrogens being female hormones. Males and females, men and women, make all of these hormones, progesterone, testosterone, and estrogens. It's just that we differ in the amount of enzymes that we have to convert one to the other. So for instance, the enzyme that converts testosterone to estrogens is called aromatase. And women have more aromatase than men, so that we can make more estrogens than men do. Okay, and also I mentioned this earlier, but there are estrogen receptors throughout the body. So of course, they're in reproductive organs like the ovary and the testes, but you may be surprised to see the wide variety of other organs in which you can find estrogen receptors. And this is why a hormone therapy, any kind of hormone therapy, can affect multiple organ systems like the lung and the heart as well as the brain. And I don't have time to go into this today, but in fact, the brain not only has receptors for estrogens, but the brain can make its own estrogens. So estrogens are not only made in the reproductive organs, but they're also made directly in the brain. Okay, the part of the brain that I'm going to focus on today is called the hippocampus, which I showed a cartoon of earlier. So the hippocampus is what we call a bilateral structure, meaning there's one on each side of our brain. And it is important for not all kinds of memory, but lots of different kinds of memories. So it's super important for things like our spatial memory or spatial navigation. So, we all have kind of a mental map of where we live, our town, our city, the campus, for instance, in our heads. And it's the hippocampus that puts all of the information necessary to assemble that map so that we can get from place to place. The hippocampus is also good about putting bunches of information together to form a context. So we learn not only facts, but we can remember where we heard those facts, who told us, where we were, what the smells were, what we were listening to. All of those things can be packaged into a memory. Hippocampus is also very good for identifying the objects in the environment. So knowing, the water bottle, the laptop, and placing them in the environment. So the water bottle is to the right for me of my laptop. So the hippocampus is very important for lots of different kinds of memory. It's very vulnerable to aging. So it's one of the first brain regions that's affected in aging, as well as Alzheimer's disease. And so it's a very important brain region from the perspective of the neurobiology of learning and memory, and has gotten a lot of attention with respect to estrogen's regulation of memory. So again, this is a cartoon of what it looks like in a human brain. In a rodent brain, it is similarly bilateral, so there's one on each side, but it extends much more dorsally in the brain. So you can see the brain from a variety of perspectives. This is an overhead view of a rodent brain. And then if you took a slice right through the middle, you'd see something like this. So this is the top of the brain, this is the bottom of the brain. And so you can see the hippocampus is much more dorsal. It's more to the top of the brain in the rodent brain. And that's true of many other species that don't have this very developed cerebral cortex. Our cerebral cortex is just so big, it's kind of shoved a lot of these subcortical brain regions down to the base of the brain, and so they don't extend as high as they used to. But regardless of that fact, the functioning of the hippocampus in a rodent like a rat or a mouse is very similar to that of a human. And so we can use these species to model what's happening in the human brain. Now, I should also mention that there are multiple kinds of estrogen receptors. The two classical forms are what we call ER alpha and ER beta. And they live generally inside of cells like neurons. And this image here is from an old paper just showing the distribution of ER alpha and ER beta in the brain. So this view of the hippocampus in the rodent is the same as this. So this is the top and this is the bottom. And along here, you can see the hippocampus, and the size of the dots represents how many of those receptors there are. So you can see that there are a lot more ER beta receptors than there are ER alpha receptors, but there are still both kinds of receptors throughout the hippocampus, and actually here in the temporal cortex that feeds a lot of sensory information to the hippocampus that it can use to package up into a memory. There is a third, more recently discovered estrogen receptor called G protein coupled estrogen receptor. I'm not going to really talk about this receptor today, but this is a receptor that lives in the cell membrane instead of inside the cell like ER alpha and ER beta. Now I'm going to focus on a specific type of neuron in the hippocampus today. It's a neuron called a pyramidal neuron. It lives in what we call the CA1 region of the hippocampus. These are big neurons that line up in these very orderly rows in the hippocampus. And they have the same sort of three major parts of a neuron that every neuron has. It is a cell body, which is roughly, these are called pyramidal cells because the cell bodies resemble a pyramid. They're roughly triangular. And the cell body of a neuron has everything that a cell body for any other cell has. So a nucleus that's got the genetic material for gene transcription and protein translation. It's got mitochondria for energy and, endoplasmic reticulum and everything that you would find in cells. But neurons are special because they have additional parts that help it send and receive information. So the receiving portion of a neuron is called a dendrite. And you can think of dendrites as trees. In fact, they're often called the dendritic trees. And you can see it from this cartoon here that all of this up here are dendrites, as well as actually these neurons, not most, but these neurons have two kinds of dendritic trees. One that extends above, we call this the apical dendritic tree, and another one that extends below the cell body, we call it the basilar dendritic tree. But you can think of these as a tree with the dendrites themselves are branches of the tree. And then hopefully you can see there's these little dots on the branches, these little specks here. Those are what are called dendritic spines. And so you can think of those as leaves of the tree. And when neurons like this connect to each other, they make those connections on the spines. So the spines serve as ways to basically increase the surface area of the dendrites and make a lot more connections with other neurons. So the more spines that a dendrite has, we think the better. It's more information that they can take in from other neighboring neurons. The third major part of the neuron is the axon, and that is right here. Every neuron has a single axon, and that is the part of the neuron that sends information out to the neighboring neurons. So the dendrites take information in, the axon relays that information out to the neighboring neuron. And so it's not shown in this figure, but at the end of this axon would be a little bulbous area we call a synaptic bouton that would connect up with a dendritic spine on a neighboring neuron to relay the information from this neuron to its neighbors. Now, what does that look like in an actual photo of those? These are silver stained neurons from our animals, and you can see the cell bodies here. Let me show you a closer view. You can see the nice cell bodies that are triangular here, these pyramidal cell bodies. Here you can see a single axon coming off the base of that neuron. And then you can see the dendritic tree. And you can hopefully see these little tiny spines dotting the dendrites. It's really difficult to see those on this zoomed out view here, but you can better see the whole scope of the neuron. Now, why have I told you about this? Why do we care about dendrites and dendritic spines? We care because it turns out that these pyramidal neurons are really sensitive to levels of estradiol in the hippocampus and in the circulation. And the first demonstration of that came in the early 90s in a lab at Rockefeller University in New York City, where they found that, rodents like rats and mice don't have a menstrual cycle, female rodents. They have what's called an estrous cycle that instead of lasting 28 days, lasts only 4 or 5 days. And during the course of that cycle, they found that during the period when estradiol levels were high, we call that period proestrus, the density of these dendritic spines was a lot higher than during the periods of the cycle when estrogen levels were low. And you can see that quantified down here where green is high estrogen and blue is low estrogen. So it's about a 30% reduction in spines when estradiol levels drop. And we can see that as well if we take a female rodent, rat or mouse, and we remove her ovaries, both of them. So we do that operation is called an ovariectomy. And overectomized females have lower amounts of these dendritic spines than overectomized females who are treated with estradiol. So animals treated with what we call vehicle, just a control substance, have lower numbers of spines than animals treated with estrogens. So this is a classic finding in the field. Numerous labs, including my own, have replicated this. And in the 30-odd years since this was discovered, this phenomenon was discovered, labs throughout the world have shown that estradiol can do a lot of things in the hippocampus. I'll talk a little bit about what's called cell signaling, but we also know that it affects gene transcription, the growth of new neurons in the hippocampus. The adult brain has two places in which new neurons can be generated, and the hippocampus is one of them, and that is promoted by estradiol. Estradiol increases the growth of new neurons, generation of new neurons. Estradiol also facilitates synaptic plasticity, and as we'll see, memory as well. And as I said earlier, we think that the age-related loss of estrogens may exacerbate the effects of aging on memory. The work that we do is in a certain strain of mouse called C57 Black 6 mice, in which long ago I showed that the onset of memory decline in middle-aged females is associated with the loss of hormone cycling. So very much like in women, many women start to experience difficulties remembering, concentrating, focusing when they start going through the menopausal transition in middle age. We see that same kind of phenomenon in mice. So they're a very good model of menopause-related memory loss. And we can, very much like is seen in human women, we can improve memory in middle-aged and even aged animals with estrogen treatment. Okay, so by now you may be wondering how we measure memory in mice, because we can't talk to them and ask them what they remember. There's actually quite a number of ways in which we can measure memory in mice. The methods that we've been using in my lab in the last 10, 20 years have focused largely on asking animals what they remember about objects. So we use very simple objects that we can buy, we have bought at the campus bookstore or just a local hardware or other stores. And we put those objects into a big white box. The animals are habituated to this box because it's a new environment for them. And just to get them used to this new environment, we habituate them for several days before we start doing any kind of training. 24 hours after the last habituation trial, we then put the animals in the box with two identical objects. Now, because the life of a laboratory mouse is kind of boring, they just sit in a cage all day in the lab and do nothing. When we give them something to do, like new objects they've never seen before, they tend to be very interested in them. And so it's easy to get them to explore these objects. And we keep them in the box until they've spent 30 seconds exploring the two objects. And then for a task we call object recognition, 2 days later, we then test what they remember about these objects. So if the animals remember the familiar training objects, like this guy over here, they're not going to spend much time exploring it because they've already done that. What they should do is spend more time exploring the new object, which in this case is this little pink stapler. Because if they remember this old object, they shouldn't want to explore it anymore. Mice are inherently, and rats too, are inherently drawn to novelty. They like new things. They're curious about new things. And so they will spend more time exploring this new object over the old object. We can also, so this tells us how well the animals remember what the objects are. We can also test how well the animals know where the objects are. So we conduct another task called object placement, in which, again, the training is the same as for object recognition. The animals, just two identical objects placed near the corners of the box. Animals accumulate 30 seconds exploring them. Here the delay is a little bit shorter, 24 hours. But during testing, here instead of putting in a new object, we simply take one of the training objects and move it to a new place. And remarkably, they are smart enough to notice this change. If they remember the location of the objects during training, they're going to spend more time with the moved object than the non-moved object. So the key here is what happens during testing. where the object recognition task allows us to test what the animals, if the animals remember what the objects are, and the placement task allows us to test where the animals are or where the objects are in space. And this is an example of the kinds of data that we get from this task. So let me explain the graph to you. 30 seconds is the most amount of time that the animals can spend with the two objects. We have time spent with objects on the y-axis. 30 seconds is the maximum time they can spend with the objects. 15 seconds is what we call chance. That means the animals are spending the exact same time with the two objects, so they don't remember the training objects because they're not spending more time than chance with the familiar training objects. These animals here, these are what we call vehicle animals. These animals are basically your equivalent of a placebo. They get a saline treatment, and that's our control. On the other hand, animals where we infuse or inject estradiol directly into the hippocampus, those animals do show memory for the training objects because they are spending a significantly more time than chance or than the vehicle treated animals with the novel object in the object recognition task and with the moved object in the object placement task. So we consider this enhanced memory in the estradiol treated animals. The animals are remembering the identity and location of the objects much longer than they would if than without estradiol treatment. And we have seen this effect dozens and dozens of times over the last 20 or so years. And so we can use this effect as a way to probe underlying mechanisms. So we can say, how does estradiol enhance memory? Okay, we know it does. How does it do that? So that we can learn about ways in which we can, potential targets for drug development. So without getting into too much detail, there's a couple of ways in which, sort of broad classes of ways in which estradiol can do that. The classical mechanisms would be for estradiol to bind to ER alpha and ER beta that are inside the cell. This here represents the cell membrane, the lipid bilayer membrane. And estradiol, if you remember, is from cholesterol to lipid. So it very easily moves through the membrane, binds to its receptor. And then that receptor hormone complex binds to DNA in the nucleus and leading to gene transcription. And that may account for some of our effects on memory, but probably not many, if at all, because the effects of estradiol on memory are rather quick. And so we think that these non-classical mechanisms where ER alpha and ER beta are acting not in the nucleus, but actually at the plasma membrane in combination with what are called neurotransmitter receptors, like glutamate receptors, help to trigger some rapid intracellular changes in things like cell signaling and ultimately gene transcription, but perhaps we'll talk about this today, some protein translation, that are probably mediating the effects that we're seeing on memory. And I put this big scary figure this is our current model of the molecules that we have so far shown are necessary for estradiol to enhance memory. And I'm not going to go into this in any detail, but just to say that the receptors molecules in purple and blue, these are receptors that we've shown are important for estradiol to enhance memory. What you see here in green, these are enzymes that are cell signaling enzymes. Cell signaling molecules, these enzymes are like dominoes falling. So you trigger one, and that usually by adding a phosphate group to the enzyme, that's called phosphorylation. Phosphorylation of one, say pKa, then leads to phosphorylation of the next down the chain, and the next one, and the next one, and the next one, and so on. So it's like dominoes falling. The particular, and that usually leads to something in the nucleus, like gene transcription, which you see here in orange. The kinase that we initially became interested in, and that I'll highlight today, is called ERK, or extracellular signal regulating kinase. So this is an enzyme that when a phosphate group is added to it, becomes phosphorylated, and that causes it to do a number of things. It can affect gene transcription directly. It can affect it indirectly through epigenetic changes. I'm going to focus today on its role in protein synthesis. But back when I was still at Yale, we became interested in the role of ERK in memory because we found, well, a variety of literature had shown that ERK was needed to be phosphorylated in order for animals to form long-term memories. And some other data suggested that estradiol could play a role in phosphorylating ERK. And so we conducted a couple of studies in young and middle-aged females to see if ERK might have a role in estrogen's effects on memory. And in fact, we found that it did. And this is ERK within the hippocampus. What I'm showing you here is something called a Western blot. It's a way for us to look to see how much protein of a certain type is in our sample of interest. In this case, we were interested in this phosphorylated, this activated ERK protein. And when we infused estradiol into the dorsal hippocampus or into one of the fluid-filled spaces of the brain called the dorsal third ventricle, we saw an increase in the activation, the phosphorylation of this ERK, and that could be blocked by a drug called UO126 that prevents ERK from being phosphorylated. And it turns out that UO126 not only prevents estradiol from phosphorylating ERK, but it also prevents estradiol from enhancing memory. So this is the object recognition task. Again, 30 seconds with the objects, 15 seconds is chance. Animals receiving estradiol treatment show enhanced memory. Animals receiving estradiol plus this ERK inhibitor do not. We then wanted to ask, we've looked at a number of things that lie upstream from ERK. Today, I'm just going to talk a little bit about the estrogen receptors that might mediate the estradiol's effects on ERK and memory. So we became interested in ER alpha and ER beta signaling at the membrane in collaboration with this metabotropic glutamate receptor. And I'll just tell you a little bit about how we looked at that. We used drugs called agonists. This was work done by a former postdoc in the lab, Marissa Boulware. And Marissa infused into the dorsal hippocampus a drug called PPT and another drug called DPN. PPT is an ER alpha agonist. So an agonist drug is basically a mimetic. It's a drug that binds to the receptor and acts like its natural ligand, which in this case is estradiol. So PPT binds to ER alpha and activates it just like estradiol would. DPN binds to ER beta, just like estradiol would. And then she looked at the effects of PPT and DPN on their own on memory, and then what would happen when she combined those drugs with this ERK inhibitor. And so this is a lot here. I'm just going to point out the most relevant pieces. So we have our two tasks, object recognition and object placement. And you can see here that PPT and DPN on their own both enhance memory, just like estradiol. So activating either ER alpha or ER beta enhances memory like estradiol does. And that effect, the effects of both of those drugs are blocked completely by UO126. UO126 also prevents those drugs from increasing ERK phosphorylation. So in a nutshell, these data tell us that ER alpha and ER beta, and I'm not showing you data demonstrating that there's an interaction with this receptor, but just suffice to say that ER alpha and ER beta can enhance memory by activating ERK. Now, downstream from ERK, ERK is involved in regulating protein synthesis. Now, we thought this might be important because of estradiol's effects on spines. In order to make a spine, any kind of structural change, you need new proteins. And it turns out that in dendrites, dendrites have an ability to make proteins within the dendrites that's completely separate from the cell body. And so we thought that this what we call local protein synthesis within dendrites might be important for estradiol's effects on memory. And that local protein synthesis is turned on by another kinase called mTOR, which stands for mammalian target of rapamycin. And ERK phosphorylation triggers mTOR phosphorylation, which then leads to protein synthesis, and we thought to spines. So we first looked at the protein synthesis, sort of the mTOR portion of this, and then looked at spines. So with respect to the role of mTOR, another former postdoc, Ashley Fortress, in the lab at UWM, looked to see whether an inhibitor of mTOR, rapamycin, could block the effects of estradiol on ERK and on memory. And in fact, it did. So then later we were able to look to see whether activation of ERK and mTOR were necessary for estradiol to increase those spines that I showed you earlier. So these are micrographs of actual spines from our animals. So these are animals treated with a vehicle control, and animals, these are overactomized female mice treated with estradiol. And you can see a lot more of these spines in the estradiol treated animal than in the vehicle treated animal. And again, these are those CA1 pyramidal neurons. And so this work was done by a former graduate student in the lab, Jennifer Tuscher, and Jen found that indeed activation of those two cell signaling enzymes were necessary for estradiol to increase spines. So she infused estradiol into the dorsal hippocampus and looked at those apical and those basal spines. And I'm just showing you data from basal spines because the data is identical for both spine types. So she found an increase in spines within 30 minutes. So that's pretty rapid, and it lasted for 2 hours. But then if she combined, so you can see that again here for estradiol plus vehicle, you see an increase in spines relative to vehicle. But if she co-infused estradiol with inhibitors of ERK or mTOR, those effects went away on spines, telling us that this rapid cell signaling was necessary for estradiol to increase spines. And that was actually very exciting at the time because we'd known for 20, 30 years that estradiol regulated spines, but we didn't know how. So this was the first study to actually show how that regulation is happening. So for our purposes, this is a very simplified version of how estradiol might enhance memory. But the data that I've highlighted today show that estrogen receptors, ER alpha and ER beta, can increase ERK phosphorylation, which leads to activation of mTOR, which leads to protein synthesis and leading to new spines and improved memory. So you might be asking yourself, wow, okay, that's fine, but so what? How would that lead to new drugs? What does this have to do with drug development? Well, we go back to this initial sex difference, trying to understand, if we can understand the basic mechanisms through which estradiol is regulating memory in younger animals, we can then perhaps some of those molecules that we identify are important for estradiol to regulate memory. Those could be targets for drug development that we can either prevent or reduce women's risks of developing age-related memory decline, memory dysfunction during menopause, or even potentially Alzheimer's disease. So, and another line of research in the lab that's related to this basic research, we've been trying to address some of the symptoms of menopause. Obviously, we're very much interested in memory and cognitive dysfunction that happens during menopause, but there are lots of symptoms of menopause, many more than many people, I think, recognize. Of course, we're all familiar with hot flashes. That's commonly associated with menopause, but there are also mood changes like increases in depression and anxiety. There are other sort of reproductive changes like irregular periods, obviously, vaginal dryness, some incontinence, and also weight gain. And a lot of women are bothered by this seemingly uncontrollable weight gain as they get older. And so hormone replacement therapy, as I mentioned earlier, has some benefits particularly for hot flashes and mood swings. Also can be very useful for alleviating vaginal dryness and for difficulty concentrating. But as I mentioned earlier as well, there are risks, particularly breast and endometrial cancer. And it turns out that of the two estrogen receptors that we've been talking about, it's ER alpha that causes these side effects, not ER beta. In fact, ER beta is associated not with an increased risk of cancer, but a decreased risk. It's actually anti-tumorigenic. And several years ago, I started collaborating with Dr. William Donaldson at Marquette University and Dr. Daniel Sen at Concordia University, Wisconsin, to develop new drugs that could selectively target ER beta, because ER beta seems to have a lot, responsible for a lot of the benefits of estrogens without some of those side effects. So as I just showed you, ER alpha activates ERK and mTOR signaling to increase hippocampal dendritic spines, and it also enhances memory. Now, ER alpha can do that too, but ER alpha is associated with these detrimental effects of hormone therapy, whereas ER beta has anti-tumor properties. It also, in numerous models, relieves hot flashes and improves mood. So it's got a lot of things to like about it. So Bill, he's a synthetic chemist at Marquette, and he and his students have developed a whole library of different ER beta compounds that are agonists. So again, an agonist is a drug that's going to mimic the endogenous drug. In this case, the compound, the agonist, will mimic the effects of estradiol at the ER beta receptor. And our lead compound is something we call EGX358, and it has a pretty staggering 750-fold greater selectivity for ER beta than for ER alpha. And the molecule itself is highlighted here in red. So this is the molecule laid over top of estradiol. So you can see that it's obviously very structurally related to estradiol. It's sort of a pared back version of estradiol. And it's very selective for ER beta. There's one other estrogen receptor beta agonist that's currently in clinical trials, and it's only about 32-fold more selective for ER beta than for ER alpha. And so it's going to have significantly greater risk profile than our compound would. Now, we've done some studies in mice with this compound. Our first was done by another former graduate student of mine, Dr. Jae-Kyun Kim, who administered EGX358 to young adult overectomized mice in a variety of different ways. He infused it directly into the hippocampus, he injected it into the abdomen, and what I'm showing you here is he administered via oral gavage. We kind of stick a blunt needle down into the stomach and inject it directly into the stomach. And what you see here are the result of, we tested the animals in both the object recognition and object placement tasks. The results were the same for both of them. Here again, you see 30 seconds spent with the object's maximum, 15 seconds is chance. Our vehicle-treated control animals are spending chance amounts of time with the objects. This is DPN, the ER beta agonist I showed you earlier in the talk. Oops. Still enhancing memory in these later experiments like it did before. But now we have a couple of doses of our new compound, EGX358, and showing that enhances memory as well as DPN. We can see this enhancement when we administer EGX via a single dose, or whether we give it every day for a couple of months. What's nice about the compound is it also has no effects on breast cancer cell proliferation. It doesn't bind to other nuclear hormone receptors. So it's really specific to ER beta. It has no gross effects on tissue pathology or on CBC blood panel. And then most recently, one of my current students, Aaron Fleischer, expanded our test battery beyond just memory to also look at some other menopausal symptoms, hot flashes, as well as depression and anxiety-like behavior and body weight. So you might ask, how do you measure a hot flash in mice? Again, we can't ask them if they're experiencing a hot flash. So the way that we do that is by infrared imaging of their tails. So mice cannot sweat to release heat, and they can't pant like dogs. They release heat out their tails. And so we can take pictures with infrared camera to see where the animals are dissipating heat from. And this top figure here, you can see an animal at rest. This is their tail outlined here in the dotted line, and they're not releasing heat. This is a vehicle-treated animal. The animal down below was injected with a compound called sanctide, which acts in the hypothalamic parts of the brain to increase temperature. And here you can see a very hot tail. This animal is releasing a lot of heat from their tail. And Aaron injected 4 groups of mice with this sanctide to induce a hot flash. And several things to notice here. The hot flash onset is pretty sudden. Within a couple of minutes, we have sort of peak increase of several degrees of temperature, and it's short-lived. So by about 10 minutes or so after injection, we're back, pretty close to baseline. But also animals, so vehicle-treated animals are in black, and animals treated with estradiol, DPN, or EGX are in the colors. And you can see that all 3 of the estrogen treatments reduces the magnitude of that hot flash. So we're pretty excited about that, suggesting that EGX could potentially be used to reduce hot flashes. Now, initially, we were kind of disappointed that we didn't see any effects on depression or anxiety-like behaviors or on body weight. But also we didn't see any negative effects of EGX on any of those things. And so while we were hoping to see some benefits on those other parameters, we didn't see any negative effects on those parameters. So treatment so far would suggest that we would have no adverse effects on mood or on body weight. So what are our next steps? Oh, I should also mention, I keep at the top of the screen, it says Estrogen Therapeutics, but I haven't mentioned what that is. So back in 2018, Bill, Dan, and I formed a company to develop these compounds called Estrogen Therapeutics, Inc. And so we are currently working to complete some additional preclinical studies. We are submitting, actually next week, a small business grant to the NIH to help us do that. Then we plan to file an investigational new drug application to the FDA to allow us to do phase 1 and phase 2 clinical trials. And then the ultimate goal is to license the compounds to a pharmaceutical partner. Now, we are working to do this in conjunction with all 3 universities. So the UWM Research Foundation, Marquette, and Concordia have been enormously supportive of our work, and we work very closely with their patent offices. We have 2 patents pending on these compounds. The UWM Research Foundation in particular, Brian Thompson, Jessica Savaji, and their colleagues have been enormously supportive of the work that we are doing, for which we're eternally grateful. So they've supported not only preclinical research on this project, but they've also been providing us with seed funding for the business itself, as well as mentoring through numerous programs and support in terms of training us on entrepreneurial activities. And so the Research Foundation has been just fantastically supportive of this project. Okay, so at that, I'll end and take some questions. I just wanted to first acknowledge the hard work of the people in the lab who do this work. So this was the lab the last time that we were able to all be in the same room. This was a holiday party we had in 2019. And that's the last time I saw everybody in person, all at the same place. We've been back in the lab since the end of July, but everything's virtual. Many of our interactions are virtual. There are students, all the graduate students and postdoc are back in the lab. They rotate with social distancing and rotating who's working in what space so that they don't infect each other. But although everybody's almost fully vaccinated. So, but anyway, I want to thank the contributions of current and past postdocs, graduate students, undergraduates, as well as our collaborators, especially Dan and Bill, and our funding sources. And at this point, I'm happy to stop and take any questions you might have.
Thank you very much, Karyn. That was fascinating. A couple questions have come in, and I encourage, we have about 6 minutes left. If there's any other questions, please put them in the chat. But there's one from Ken. He asked, would testosterone be considered protective related to memory loss?
Yes, actually testosterone would. There's not nearly as much information. There just hasn't been as much research done on testosterone as there is estradiol, but there is data to suggest that testosterone can be beneficial for memory, although it's unclear how it's being helpful, whether it's acting as testosterone or whether it's acting as it gets metabolized into estradiol and then has effects on memory. But yes, not only testosterone, but also progesterone can be beneficial for memory as well. And one thing that I didn't mention, I didn't have time to get into it, but estradiol can be actually very beneficial for memory in males as well. And so many of the same things we see for estradiol in terms of spines and cell signaling and memory happens in males as well as females. There's just not as much work that's been done so far on males.
Okay, well, you answered the next question in that one, so well done. And then we have, can you talk a bit more about the work being done in this field with Alzheimer's disease?
Yes, absolutely. There are a number of fronts in which people are looking at estrogens and Alzheimer's disease. One of them that we actually happen to be involved in is trying to understand how various genetic risk factors intersect with estrogens and estrogen loss. And so without going into too much detail, in addition to age and female sex, there's one genetic risk factor called apolipoprotein E that is associated with late onset Alzheimer's disease, which is the more common form of Alzheimer's. This would strike people in their mid-60s and beyond. And a certain gene variant of apolipoprotein E called E4 puts you at much greater risk of developing Alzheimer's disease. And in fact, women who have 2 copies of this E4 gene are at greatest risk of developing Alzheimer's relative to men who have 2 copies or men and women of any of the other gene variants. And so we're trying to figure out why that is and whether we can potentially reduce the memory dysfunction that's seen in females with this E4 allele with estrogen therapy. So there are, like I said, a number of different labs throughout the world who are trying to figure this out. There are Roberta Brinton at Arizona State is doing a lot with estrogens and metabolism in the brain and seeing some interesting effects showing that women's the metabolism in the brain is affected by menopause and hormone loss there, and that increases women's risk of Alzheimer's disease. So if we potentially reverse those metabolic changes, we can maybe decrease women's risk.
Okay, and we have another question here from Karyn. What's the time frame? How many years before this might make it to market as a pharmaceutical?
Oh, that's a really good question. So I would say optimistically, maybe 5 to 10 years, probably more along the 7 to 10 year. I don't know. It's hard to tell. I mean, right now it probably will take us another couple of years of preclinical work because it's not the work proceeds relatively slowly, but applying for the funding to get the work and then having the applications go through the review process at the NIH takes time. So the application that we'll submit in April will be reviewed in the fall, so probably September, and then we won't know whether we'll receive funding until probably late 2021, maybe early 2022. And that, so it's a good 9 months or so before we would even get the funding, and then we'll do the work. So unfortunately, things take a while, in part due to the federal review process. But but hopefully all goes well and we're able to get this product to the market, I don't know, within 7 years or so.
All right, fingers crossed.
I know.
Then a final question from Katie to round us out today. Have you done research on brain injuries and how that affects memory loss?
I have not, but I have colleagues. A good friend of mine at Texas A&M Health Science Center has done work on brain injuries. And so there are some interesting, estrogens seem to be protective for brain injury at certain points of a female's life cycle, but not others. And so she's trying to understand why women tend to suffer more from stroke. When they do have an instance of stroke, the damage tends to be more severe. And it seems that's particularly true in postmenopausal women. And so there's do seem to be interesting interactions between estrogen loss and magnitude of damage due to stroke. So there are people out there doing that, but that's not something we focused on.
Okay. We are right at our time, so I don't want to take more because I know time is precious these days. But thank you so much for sharing your research and your expertise with us. You know, we have alums from all over the place that have tuned in, and we'd love to have you live on campus sometime when we get back to normal world lectures.
It would be my pleasure. It would be my pleasure. Thanks everyone for taking some time to spend with me today.
Thank you again. It was our pleasure.